Thank you, Anna. Good morning, everyone. Good afternoon, everyone. I'll try to be brief, as I realize that I am the only thing that is avoiding some of you going to lunch. I'll take a couple of minutes just to introduce you our center, the Center for Translational Research in Oncology at Instituto do Câncer do Estado de São Paulo. It's a newly formed institute. It's 7 years now, and our laboratory was established there 4 years ago. At our institute, we have 4 major research interests. We are working in clinical research in molecular oncology, innovative therapeutics, and epidemiology and prevention. Our focus in clinical research are drug-targeted therapies, pharmacoeconomics, and the incorporation of new technologies, our universal access system of health in the country. In molecular oncology, we work with molecular epidemiology, biomarkers, and clinical application. Molecular imaging is a very dear subject for us, and systems oncology. In the innovative therapeutics, we are addressing chemosensitization strategies and working with gene therapy and immune therapy. And in the epidemiology prevention group, we have cohort studies and vaccination protocols, especially aiming at the control of papillomavirus associated diseases in the country. The way we organize our institute, it's as a network. Indeed, we have at the University of São Paulo, we belong to the University of São Paulo. We have around 70 or so groups working in cancer research. We are 7 groups resident at the Cancer Institute, but our commitment is to interact with all cancer research groups at the University of São Paulo and in other centers as Camargo Hospital, for example, and Syrio-Libanese, who is here represented by Ana Maria. We have different platforms, a biobank, a recombinant DNA expression and vector construction, sequencing, cytometry and epidemiology, core labs, and we interact with other groups at the medical school and at the University of São Paulo. So we are trying to develop a platform for ongoing studies in cancer research, which now receives about 6 to 8% of all funding in the state of São Paulo, at least by this year. Last year received 6% of all funding from FAPESP. So our vision is that we are created as the interface between the Secretary of Health and the Cancer Management Network in the state of São Paulo that ICESP supervises, or it interacts with 71 oncology centers throughout the state, and at the same time that it has this cancer management task, we also envisage that at the research level, our laboratories at ICESP will will interface the Secretary of Health of the Academic Cancer Research Network, which is a very, very broad virtual institute of cancer in the whole state of São Paulo, and especially at the University of São Paulo. So the subject I would like to talk today is something that is undergoing in our laboratory, and it illustrates an active interaction we have with University of California in Davis, mainly through the group of Futon Liu, who was at the Department of Dermatology. Now he's at Academia Sinica in China. And it was funded by FAPESP and by a consortium for functional glycomics from NIH. And many talented people in my group had been involved with this initiative. And later on, Emerson Soares Bernardes, who is now an independent researcher at the IPEN, and his group. IPEN is an institute for nuclear energy here in São Paulo. We worked together to try to understand the functions of a specific molecule that happens to be a cancer marker. This molecule is a member of a family of very ancient molecules, galactins. They are lectins, they interact with carbohydrates, and the molecule we are interested in is galactin 3. is a hybrid molecule. It has an activity of interacting with carbohydrates, and it has a domain in the amino terminus domain that interacts with extracellular matrix. So it was supposedly, this molecule was supposedly to be an extracellular matrix, but through the time we discovered it was not such a thing. It was not an extracellular matrix protein. It was something that would disrupt the interaction of molecules with the extracellular matrix. We have pretty much information on how galactins interact with the carbohydrates, and together with Photon Liu had developed an animal system, a galactin-3 deficient system, and through the consortium we could exploit what were the functions of galactins specifically in the immune system. And we are focusing at that time in monocyte macrophage functions, in the organization of granulomatous inflammation, and also in tumor-associated angiogenesis. In a nutshell, galactin-3 is responsible for polarizing the response towards M2 phenotype as a pro-tumorigenic phenotype, and M2-Th2 inflammation or granulomatous inflammation. So in the absence of galactin-3, what we observed in general was that there was an attenuation of M2 and Th2 responses towards M1 and Th1 pole. We observed that galactins were not only responsible for monocyte macrophage interactions, but also galactin was very important in B cell and plasma in B cell differentiation. In the absence of galactin-3, we have accelerated plasma cell differentiation, plasma cell maturation, which somehow phenocopies Notch signaling. So galactins also interfere with Notch signaling. Galactin-3 interacts with Notch signaling, modifies Notch signaling. These are very important molecules, cells in the tumor microenvironment, but what we observed was that galactins were not only expressed in these immune cells in the tumor microenvironment, they could be also expressed by tumor cells themselves, and they were not expressed in the tumor cells in all tumor cells. This, for example, is glioblastoma multiformis, the stage 4 of GBM. We have a very specific architecture here in glioblastomas. This we call pseudopalisades. These are tumor cells which are running away, if you will, from necrotic area. And these cells which are surviving in these environments and running away from necrotic area express larger amounts of galactin-3. And when we first saw this data, we start asking if galactin-3 would dictate the running of the cell out of the necrotic area, or it will inject a death signal and will make these cells in the palisade transforming in necrotic cells, as we find here. So we postulate that galactin-3 either would dictate a run or a die sentence to the cells. In order to figure out that, we established model systems where we could recapitulate the overexpression of galactin-3 in the pseudopalisading areas, and observed that was associated with viable cells that are in hypoxic areas in the tissues. We went towards the mechanism, and I will run through these slides because of time constraints. So what we observed was that under hypoxia and the privation of serum, galactin-3 will go up, and expression will go up in these tumor cells, and this was somehow related to specific signaling dependent on one transcription factor, HIF, the hypoxic-inducing factor, and another inflammation-associated transcription factor, NF-kappa B. So both NF-kappa B dependent transcription of galactin and HIF transcription-dependent of galectin-3 were taking place when tumor cells were exposed to serum deprivation in hypoxic. We could show that this was the mechanism behind, and the function of galectin-3 was to protect cells from cell death, and we could get to this conclusion when we inhibited the expression of galectin-3 using specific siRNA against galectin-3. In the absence of galectin-3, cells die more. So if we now prepare one cell that is blocked, has galectin-3 expression blocked, we don't see the frequency of tumors is altered. So galectin-3 is necessary for a tumor cell to develop into a tumor in an animal. If it has no galectin-3, we will see tumors, but much later on. So galectin-3 seems to be a very essential molecule for glioblastoma cells to survive and generate all the tissue changes that are necessary for the cell to grow as a tumor. In the sense, so it will make cells survive, and eventually it gets secreted. And when galectin-3 gets secreted, galectin-3 may now interact with extracellular matrix molecules, and there on the extracellular space, galectin-3 will change the cytoarchitecture of the cell and organize this lamellipodia, which is very common features of migrating cells. So once in the extracellular matrix, galectin-3 would prompt cells to migrate. Indeed, we could see that again using the galectin-3 cells derived from galectin-3 null mice and cells derived from wild-type mice. Cells which are derived from wild-type mice express galectin-3. They adhere less than galectin-3 null cells, and they migrate much faster. So cells which have galectin-3 are less adherent and more migratory than cells which do not express galectin-3. If we restore the expression of galectin-3 in galectin-3 null cells, we invert this phenotype. So in the presence of galectin-3, cells would adhere less and would migrate more in a way that could be inhibitable by carbohydrates. So it's a lectin function of galectin-3 that is prompting surviving cells to migrate. And this is dependent on the arrangement of the focal adhesion plaques, decreasing the focal adhesion kinase, and signaling through AKT, which gets readily phosphorylated when we incubate cells with extracellular galectin-3. So in the first level, galectin-3 would provide signals to survival. If galectin-3 expression is maintained high, cells will migrate out of a certain microenvironment. And if we get galectin-3 levels up in the tissue, it will lead to other alterations in the tissue. And there we could see that using a model that we developed in our laboratory and we further characterized with Ana Maria Camargo at the Ludwig Institute of Cancer Research, the melanoma cells that are devoid of galectin-3 expression through complete epigenetic silencing of its promoter, so it has its promoter hypermethylated. So we had melanoma cells that lacked the expression of galectin-3 through silencing of the gene. So we had cells that express galectin-3, the cell that does not express galectin-3, and we challenged animals which had galectin-3 and in animals which did not have galectin-3. So we have 4 situations, and what we observed was that when we have galectin-3 either from the tumor microenvironment or from both tumor microenvironment and tumor cells, the tumors grow much faster and larger tumors appear. Then we compare with those situations that we have neither galectin-3 from tumor cells nor the tumor microenvironment, and when we have an intermediate situation, tumors grows to an intermediate level. In that sense, galectin-3 seems to prompt a signal that would enable tumors to grow, and this signal is associated with sustained angiogenesis, so sustained expression of galectin-3 would maintain the formation of vessels. If we don't have galectin-3 in tumor cells or in both tumor cells and the microenvironment, we lose galectin-3 expression, and that was clearly associated with signaling from VEGFR2 receptor, which is the receptor is necessary for angiogenesis in most of the biological systems we know of so far. This is associated with altered macrophage again, infiltration, and so it's extracellular function of galectin-3 which seems impaired here, and if so, it's potentially targetable. So we could target galectin-3 in the extracellular space and try to avoid tumor growth with different strategies. And one possible strategy for that is to use pectins, which are molecules that interact with galectin-3. If we remove galectin-3 from the system using these pectins, we get decrease in tumor growth. The question is, when we do this combination, we don't know. And we know at the pathophysiology we can have some clues about the role of these molecules, but then the issue is when we do the intervention. And to get that, we need to have real-time information, as much real-time information as possible. And we started to realize that, and then we organized projects to understand in real time what would happen with the tumor microenvironment and when it's the right time to intervene in terms of having, for example, modifying galectin-3 expression. So imaging is for us a priority now in the laboratory, and we started that by studying aspects of angiogenesis and vascular function control. And we are now moving forwards. I will show you briefly two sets of data we have. One is associated with a molecule that inhibits the angiotensin system, which was already mentioned today. There are molecules that block the angiotensin receptor. Angiotensin receptor would signal an increase in vascular tonus. And there are antihypertensive agents such as losartan, for example, or candesartan, that block the AT1 receptor, and doing so, they would decrease the vascular tonus, leading to hypotension or normalization of pressure. Something we observed a couple of years ago was that in tumors, we have an overexpression of AT1 receptor in the vascular system that feeds the tumor. And not only that, but we also have local production of angiotensin 2 here seen by immunohistochemistry. So tumors may produce angiotensin 2 that would interact with angiotensin 1 receptor in the pericytes or eventually in the cilia cells, in the vascular structures that feed tumors. If we block angiotensin 1 receptor function in tumors, we control tumors, we control the tumor growth, and we see that clearly using both losartan and candesartan. Recently, 3 or 4 years ago, a couple of interesting papers came and showed that patients who are receiving antihypertensives, they are at lower risk of developing cancer. So people receiving drugs such as losartan or captopril, which is an inhibitor of the angiotensin converting enzyme, is somehow protecting of developing cancer. Not only that, Rakesh Jain showed it very elegantly that when he uses losartan in very lower doses than we use in our system, he could inhibit collagen synthesis and would favor delivery of substances to tumors. And these are the experiments that they published last year or a year before, showing that if they use losartan, they normalize the tumor microenvironment, and increase the oxygenation levels of tumors. That's a very interesting concept that we are addressing in a specific system through a different way, indeed. The way we are addressing that, it was to counteract the angiotensin system using bradykinin, an observation from the bradykinin bradykinin receptor activation pathways. And this was observed using the antagonists of the bradykinin signaling, R715 and R954. And what we observed a couple of years ago was that if we treat animals with bradykinin receptor antagonists, we would have an increase in the delivery of substances to tumors. The substance here is doxorubicin. Doxorubicin is a chemotherapeutic agent used for cancer, for breast cancer treatment, for example. And if we treat animals with this vasoactive molecules, we would increase the delivery of the substances to tumors. And we would do so because of, we did so because we have uncovered an adverse effect of this bradykinin receptor antagonist, which is a transient increase in blood pressure. If we have a transient increase in blood pressure, and that was what happened with this compound here, just after the infusion of these substances, and it lasts a little bit longer than we would expect, we would have increase in delivery of chemotherapeutic agents. So we are now using this strategy to understand what substances can be used to interfere with blood tumor perfusion. This is a tumor here. You look here at one vessel with Doppler, and then we infuse one of these vasoactive substances. In this case, is a hypertonic solution. And 5 minutes after, you see an increase in blood perfusion, and this increase in blood perfusion is transient. It decreases in 10 minutes, and we are trying to use now this window here to deliver specific substances to tumors. And in order to do that, we need imaging and precision imaging indeed. So I'll stop by here. I'll just summarize this part. So I showed you before that galectins are targetable for therapy. They are complex molecules, and through imaging, and if we can address melanomas, for example, which is our model, we can add unusual drugs for melanoma treatment, such as antihypertensive agents as losartans. There is an opportunity for academic clinical trials using then antihypertensive agents to increase delivery of drugs. And there was a recently approved trial in NIH to pancreatic cancer using losartan. We are eager to see the results. And based on this scheme of angiotensin and bradykinin, we could unravel, or at least we could uncover a possible effect of these molecules that could be useful to increase delivery of substances to tumors, which would allow combination therapy, and in the right window. Indeed, this is the key issue now for combination therapy. What is the right window? Hopefully imaging will help us. So this is where we are. We are in the 8th floor here. It's this point here. You were all invited to come and visit and collaborate with us. Thank you.